Joe Sadek, psychologist, neuropsychologist at the VA, here to introduce Jeremy Hogeveen. Jeremy is in the Department of Psychology, and I'm going to bring up his bio right now. So Jeremy is an assistant professor in the Department of Psychology at UNM. Before joining UNM, he completed a Ph.D. in Cognitive Neuroscience at Wilfrid Laurier University in Ontario, Canada. In postdoctoral training in both Cognitive Neuropsychology at Northwestern University with Dr. Jordan Grafman, and Developmental Psychopathology at the UC Davis Mind Institute with Dr. Marjorie Solomon. Jeremy's research aims to use brain imaging to understand the normal functioning of neural circuits underlying motivated decision-making, their role in health and disease, and the potential impact of targeted non-invasive stimulation of these circuits. Looking forward to this presentation, Jeremy. Thank you for presenting today.
Thank you very much, Joe. I'll go ahead and share my screen. Can everybody hear me okay? We could get some thumbs up. Anybody?
We can hear, yes, thank you.
Great. You'd assume that I would know that that's always working, given I've been doing the Zoom thing for a while now, but just want to check. So thank you all for being here today. As Joe mentioned, my name is Jeremy Hogeveen. Hopefully you see the slides and not presenter view. Can I get a thumbs up for that one too?
Yeah.
Okay, great. So the title of my talk today is Neural Mechanisms of Motivated Choice. And the little brain map I have there on the slide, I'll present some data from it later on today. It's basically it was a lesion symptom map that we did a number of years ago now that kind of set me down the path to studying this topic. So it seemed fitting to put it up there on the title page. Before I get started here, I just need to acknowledge the big community of collaborators and colleagues that have helped me in some way to present some of the work that I'm doing here today, as well as obviously the grad students and the research assistants who do the real work and help me get the data so that I have something on my slides here, as well as funders who have helped to support the work. So before I get started, I wanted to introduce you to the sort of general theoretical orientation for a lot of what I work on these days, which is kind of grounded in the computational psychiatry framework that's recently gained favor at NIMH through the leadership of Josh Gordon, et cetera. And the basic idea is that we want to use tools from computational perspectives, whether it's computer science or data science, in order to better understand and predict mental illness. A lot of folks have taken kind of a big data approach to computational psychiatry, where you have large data sets, whether it's through the ABCD collective that's been collected by the NIH recently, or other large consortium type data sets, and use machine learning and other types of algorithms to predict psychiatric cases versus control participants based on clinical variables, demographic variables, brain variables, what have you. And it's kind of agnostic to underlying mechanisms. The basic idea is you want to use the data to predict what a clinical outcome is going to be in order to guide treatment or more effective treatment targeting. And so that's one general perspective in computational psychiatry that has grown a lot recently, especially with bigger computing resources available to do those types of studies. Where our work fits in and the perspective that we tend to take in our lab is a little bit more, I tend to call it like artisanal than that approach. So we collect data in our own laboratory and test theory-driven models of neural mechanisms within subjects. We still do some of that big data stuff, but the more straightforward approach that we do is to use computational principles to understand normative functioning of the brain in order to better understand what might be abnormative or aberrant in folks that have psychiatric or neurological disorders. And this can help us not only to understand the clinical population better, but also to potentially better understand the brain itself. So by looking at what can happen with when a specific neural circuit goes wrong, we actually have kind of a causal demonstration of that circuit's involvement in typical, whatever that is, typical functioning. And a good example of this type of approach is the recent predictive coding accounts of psychosis that have been advocated. But yeah, just to state it one more way, just to make sure it's clear, you have neurobiological mechanisms that neuroscientists study, clinical phenomena that clinicians study, and with computational psychiatry and computational neuroscience, we're trying to use computational operations to build a bridge between those two, essentially. So that's what I'll talk about today with most of the work I go over. And I will be taking that approach to the study of psychiatric symptoms in folks that have had a brain injury initially. Later on, we'll talk a little bit about a neurodevelopmental example of looking at things this way, but I think a lot of the work I've been doing at UNM since I got here, thanks to a COBRE that some of the folks on the call, Doctors Quinn and Jessica Richardson and Dr. Rick Campbell as well, have all helped me get involved in, was a brain injury COBRE here at UNM. So we're going to talk about some of the data that we collected through that. So this is a lesion symptom overlay of some folks who have a traumatic brain injury. As you can see, the majority of them have sort of prefrontal cortices that are affected by their lesions. And what you tend to see clinically in these folks after their brain injury are increases in psychiatric comorbidities. So traumatic brain injuries can often be thought of primarily in terms of their motoric or cognitive deficits, but there are definite emotion and decision-making challenges that a lot of patients will face after a brain injury, especially brain injuries that affect prefrontal regions like this. And one of them that we've studied quite a bit in my lab, both since I came to UNM and before that when I was a postdoc, is apathy in TBI. So with apathy, you can kind of think of a neurological homologue of something like anhedonia or anhedonic depression in a psychiatry. I realize I'm talking to a psychiatry department. I just want to reframe things in a way that makes sense across the gap there. So when we think of apathy, we're talking about patients who had a level of motivation and goal-directed behaviors that, let's say, was up here, and then they had an injury, whether it's a car accident or getting a blow to the head in some other way, or perhaps a stroke, or maybe onset of a neurodegenerative disease that leads to a change in motivation where there's a reduction down to some post-injury state that suggests that they have a difficult time getting motivated to initiate goal-directed behaviors. And so that's something that can really affect patients. It can seriously affect their caregivers as well. As you can imagine, if you're caring for somebody who has a brain injury, their degree of apathy after the injury will really predict how much of a burden you experience. And this is just a meta-analysis across studies with traumatic brain injury specifically. So these would be closed head traumatic brain injury studies. So sports injuries, car accidents, that kind of thing. And you see that across studies, so meta-analytically, there's a significant effect on apathy, where apathy scores increase after a brain injury. And some of our early lesion symptom mapping work in this space found that when you take a big data set, so hundreds of patients who have had a traumatic brain injury, in this case, focal traumatic brain injuries, and you look at the statistical likelihood that somebody will have apathy if they have a lesion to a given part of the brain, you see this clear effect of ventromedial prefrontal cortex lesions on apathy. So VMPFC for short. And what I'm showing on the left here, a lot of times when I show maps like this, it tends to be brain activation. So like an fMRI image, for example, would be looking at functional activity in this way. In this case, we're looking at what's actually a lesion symptom map or a voxel-wise lesion symptom map. So each little pixel you see here that's in orange or in red on this ventromedial PFC region is actually an area where this is significant statistical increase in apathy if that part of the cortex is lesioned. And so in this case, patients that had lesions to these regions relative to age-matched controls, patients with injuries to other regions, so for example, more posterior regions like the parietal cortex, as well as patients with dorsal medial PFC, so sort of more dorsal lesions that are also in the prefrontal cortex. VMPFC lesions relative to all those other groups drove increased apathy in these patients. So some initial insight into, I mentioned the computational psychiatry idea that we go from a neuromechanism and a clinical phenomenology and try to link it with computation. So here we have the two, and down the road, we'll try to link them through the computation. So we have VMPFC lesions seem to cause amotivation or apathy in these patients. One nice benefit of having to lock down through the COVID lockdown in early 2020 was I got to collaborate with Devin Quinn and Jim Kavanaugh here in the psychology department on some data that they had collected from traumatic brain injured patients and resting state connectivity. And what we actually found was an interesting effect where that same region, so we took, in the interest of replication, we took the exact same region from this initial study back in 2017 and used it to look at resting state functional connections that were predictive of apathy in TBI, and found that there was actually a negative correlation. So increased apathy was associated with was negatively associated with connectivity between the ventromedial PFC and another part of the brain called the dorsal anterior cingulate cortex. But this correlation actually ended up being more than it appears. So you'll notice the title VMPFC hyperconnectivity rescues motivation. That's sort of a optimistic take on what's driving it. We would like to do a longitudinal study and follow this up. But the basic pitch is that if you look at the connectivity value on the y-axis here and our control group, as well as the very apathetic group with TBI, we basically see that it's anti-correlated or negatively correlated between these regions, both in controls and in apathetic TBI patients. The interesting thing here was we seem to see this tick upwards after the injury for specifically the TBI patients that did not have apathy. Suggests that this is a network that's anti-correlated or negatively correlated in the uninjured or healthy brain. And then if you have a TBI, in order to potentially compensate for some potential changes in motivation post-injury, there seems to be this ramp up or increase in functional connectivity after the injury. That was, again, based on cross-sectional data at one time point. The ideal would be that we follow somebody in stages after an injury and really see if there is this increase over time in connections between these regions. But generally speaking, across the two studies, this ventromedial PFC region, that one side of the coin, the neuromechanism side, seems to be related to a clinical phenomenon of changes in motivation after an injury. But from my perspective, this still leaves me wanting more. So having trained in cogneuro or cognitive neuroscience, one of the real questions I have is what's the reason for that linkage between this region and its potential injury to some clinical presentation? So just sketch it out schematically for you. So we have a neurobiological finding, neural circuits in VMPFC, and a clinical phenotype that clinicians would deal with, which is after a lesion to the VMPFC, folks deal with changes in motivation. And what we're trying to do a lot of the time in our studies now in the lab is understand the computational processes that link these two, with the hope that at some point understanding that better will help to titrate treatments better, help to monitor treatment outcomes better, etc. So in the interest of that, I will tell you about the main research question I've been trying to answer in my lab since I opened in 2018, which is how you decide whether to get a burger or sushi. So it's more than that, obviously. So we study something in the lab referred to as explore-exploit decision-making in the literature. And it's a sort of ubiquitous phenomenon that humans and non-human primates and rodents and other organisms that we study in the lab face when it comes to motivated decision-making. And this is the decision of, should you go to a familiar restaurant that serves your favorite burger in the city of Albuquerque, or should you go to a new sushi place that opened up near you or that was maybe recommended to you by a friend and explore whether or not you like it better than any restaurants you're currently familiar with? This is a trade-off that when we face, it's kind of difficult to understand why we would ever explore something new. So when we break it down from a computational perspective to understand what motivates humans and other organisms to make decisions, you should basically always get the burger because you know you like it. It's a safe bet type decision. There's really no negative with going back to a familiar option that has served you well in the past. However, humans tend to do a lot of exploration, especially during youth and adolescent periods, which we'll talk about later on in the talk, where we sample uncertain and potentially dangerous sometimes, or maybe disgusting when it comes to restaurants, potentially negative outcomes in order to sometimes find a new favorite and sometimes maybe be let down relative to what we could have exploited. So it's a trade-off we often face between exploiting familiar, safe bet type options and uncertain, novel options that could potentially lead to an increased future value, or maybe will bite us in the butt and we'll have something that we don't like very much. So we're really interested in the lab with how humans and other organisms resolve this dilemma, because when we formalize it, like I said, we want to take computational models to understand how the brain might resolve these types of decisions. It's very easy to understand how your brain learns to exploit. Standard operant conditioning, or in the modern parlance, reinforcement learning frameworks, will often quantify something referred to as the immediate expected value of exploiting something familiar. So you have gone to this burger restaurant in the past, and its immediate expected value went up because you liked the burger. So therefore, next time around, you were more likely to exploit it and go back and get that burger again. Or if you go to a restaurant that you did not like, that immediate expected value will go down. And next time around, you're less likely to exploit that resource in the future. So when it comes to sort of a computational or a formal mathematical perspective for how we would learn to exploit, it's pretty easy to solve. We learn through reinforcement, makes us more likely or less likely to do something again in the future. Where our perspective, I think, brings in something interesting. I was going to say valuable, but I'm talking about value in both contexts, so I wanted a better term. But something interesting or new is in the context of trying to formalize what would motivate us to explore. So we think, and others are of the same viewpoint, that humans and other organisms are basically trying to minimize uncertainty in the environment. And when we're doing that, in order to motivate us to explore novel and potentially uncertain options, we have this motivational signal called future value, or expected future value. In this case, when you have an option like a sushi restaurant somebody has recommended to you that you've never been to, or perhaps it just opened down the street from you, we think that the relative future value of exploring this resource and reducing the uncertainty associated with it is what motivates you to go try it out. And there's an actual inherent motivational signal associated with that. And this bonus parameter we can compute formally using a partially observable Markov decision process model. But in the interest of not showing a bunch of equations, from here on out, if I say IEV, or immediate expected value, you can think of that as the immediate value of exploiting something familiar. If I say the word bonus, or relative future value, I'm talking about the potential benefit from trying something new and finding a new favorite option, and the value of exploration versus the value of exploitation. And if you're making your decision and really highly weighting the value of exploiting something familiar, you'll probably always go back to the old burger restaurant. And if you're making decisions and highly weighting the value of something you might discover in the future by reducing uncertainty, then we expect you to try the sushi restaurant more often than not. And one quick point to make here, I realized when I was going through my slides just before I logged in that I don't necessarily have this slide on here, but it's worth noting. We view adaptive behavior as probably balancing these two pretty well. So not always exploiting, because then you never learn anything new, and not always exploring, because then you never really learn anything at all. You're just kind of making noisy decisions all over the place. Some balance between exploration and exploitation is likely the most adaptive type of decision profile you can demonstrate. If we see hyper exploitative behavior, you can think of that as inflexible or kind of rigid decision making. And if we see hyper exploratory behavior, we can think of that as impulsive or more noisy type decision making. So we think this gives us a framework that's formalized in this computational model for understanding that continuum of sort of inflexible to hyper exploratory, with optimal, quote unquote, being somewhere in between the two. And we can study it in the lab with a pretty straightforward task called the novelty bandit task. I'll walk through this slowly just because I think sometimes I go through it too quick and they get questions after. So I'll start off with just the initial setup of a bandit task. The word bandit comes from the sort of analogy to a one-armed bandit in a casino, aka a slot machine. In this case, we have a three-armed bandit, which just means that we're doing a probabilistic reinforcement task similar to a slot machine. So when you're doing a K-armed bandit task in the lab, in our case, a three-armed bandit, individuals are presented with three options. In our version, it could be two options, could be four, it could be five. In our case, it's three options. And we tend to choose things that initially have no inherent value. So we want things that are relatively neutral up front so we can study the brain's ability to learn value de novo in the lab. So we show people these relatively neutral images, and we assign them a nominal reward probability. In this case, it's a low, medium, or a high reward probability. So if you choose this option on the left, you'll be rewarded 20% of the time. The option on the right, 50%. The middle one, 80%. We don't tell you those nominal probabilities. You learn through trial and error learning which ones are best for you. And we also try to give people a little bit of extra motivation by saying, if you get a certain number of points in this task, we pay you an extra $20 at the end of the experiment. So we try to really motivate people to do well on the task. And generally speaking, people just, honestly, if we run it for points, people do pretty well too. Humans just like to earn points. And we do this a bunch of times where after people have learned to exploit the best available alternative, we insert a novel image and take out one of the familiar images and explicitly force the participant to face the explore-exploit trade-off. So what this is showing is every 5 to 12 trials, which is about the amount of time it takes you to figure out that these suitcases are the most valuable, we take one of the options away. It could be the suitcase, it could be the sunset here, it could be this low probability option, and we replace it with a novel option, in this case, the flowers in the middle here. And we force you to face this trade-off. Do I exploit what I know is already a pretty good option, or do I explore this novel option with the potential to find a new favorite or a new most beneficial, or maybe find one that's less good than my existing options? And because humans are noisy, we have to do this a lot of times. So in this version of the task, we have about 32 novel stimulus insertions, 224 trials. And when we do that, we get a really good estimate that tends to be pretty reliable for how likely people are going to be to explore a novel option when it's presented versus exploit a familiar favorite. And when we look at behavioral performance on this task, it looks very similar to what our computational model predicted earlier. So specifically, when a novel option pops up, people tend to explore it well above chance. So the blue line here, right after a novel option has first been presented, folks tend to explore it at an above chance level. But then over time, they learn which option is rewarding them the most, and they exploit that option. So exactly like those computational model parameters I presented earlier, the bonus parameter related to the value of exploring something novel and uncertain, that starts out high and then tapers off as you've selected the option. That's exactly what we see with choice behavior to the novel option. And then vice versa, the immediate expected value parameter that tends to scale up as you learn what's best to choose, that seems to be what we're seeing with the tendency to exploit on this task as well. So the model and behavior accord very well with one another. Also kind of interesting and a little important aside to mention is that this has tremendous translational relevance. So we collaborate with a non-human primate lab in Oregon Health Science University that does the exact same task. So the same paradigm that we do in human, they present to a monkey just with a gaze-based decision-making paradigm, although they're actually moving to a touch screen-based system soon. And when we use the same computational model to model behavior on the same task with the same reward probabilities, obviously there tends to be a few more trials presented to the monkey. I think like 900 to 1,000 trials for that first session rather than 200 like we do. But when you do that, you get the same behavior, essentially. So humans are monkeys. That's the conclusion of the talk. So essentially, when we look at this is the correlation with the model on the left plot here, and humans, 40 humans and 8 monkeys. It's notable that some of the humans actually have a really odd performance profile that correlates negatively with the model, whereas in the monkey, we have this consistent positive correlation between the two. But generally speaking, the magnitude of the correlation is the same, suggesting that our normative framework for understanding how people make explore-exploit decisions works similarly well in humans and monkeys. And also, this is a negative correlation between the bonus coefficient and IEV coefficient. Essentially, what this means is some people are explorers and some people are exploiters. So if bonus coefficient is high and IEV is low, you're really exploratory, not so good at exploiting familiar rewards. And vice versa, if you have a really high IEV coefficient and low bonus, you are a little bit more inflexible. You're kind of sticking with what's most rewarding and not really trying the novel options when you should. That negative correlation is the same magnitude in the monkey as it is in the humans. Granted, it's 8 monkeys, so a correlation coefficient is probably not the greatest, but that's actually a lot for a monkey study. The main reason that I show this, and I think this is interesting, is because at some point in the future, if we ever want to do sort of circuit-specific manipulations with things like optogenetics or DREADS in the monkey and compare the effects to something like TMS stimulation in the human, we can actually just compare it on the same task. If we're ever doing treatment design studies and testing the effects of new drugs on explore-exploit behavior, they can be tried out with animal models that are exactly translatable to the human rather than very indirectly associated with the human, like you would do in a rodent study. So what have we discovered from studying this in the context of neuroimaging in our lab? I have lots of brain images from this paper that I'm citing here, but I'm trying to summarize it all in one easy to digest figure. So hopefully this works. So we have kind of three different patterns of results. See some areas in blue, some in red, and then one really interesting area over here in purpley pink. So in the red areas, the ventromedial PFC, which we linked to apathy earlier on in the talk, as well as subcortical motivational circuits like the amygdala and the ventral striatum, they seem to encode positively both the IEV and the bonus parameter, suggesting that they encode the value of exploiting something familiar as well as the value of exploring something new in the same direction. What that suggests to us is that those are getting kind of a summed action value signal associated with that option, whether it's to explore or to exploit. Whereas what we see in these blue regions, so the dorsal striatum, dorsal medial PFC, and dorsolateral PFC, is this interesting flipped sign encoding. So they positively encode the value of exploring something new. So when you're making the difficult choice to try something novel, activity in these regions ramps up. However, they negatively encode the value of exploiting something familiar. So on trials when IEV is high, suggesting you really already know what you're going to do before you go in, therefore the effort associated with the decision is relatively low, these areas decrease their activation. So in these red motivational regions, we have this kind of general motivational signal that's in the same direction, regardless of what you're doing. In blue, we have regions that seem to be more associated with a difficult choice to try something novel that could pay off later, that deactivates when you're doing something easy and sticking with kind of a habit-based exploit decision. The really interesting area, the area that I could see myself spending the next like 10 years of my career focused on, is the lateral frontal polar cortex. This area does not care about exploiting. It only cares about exploring something novel. And I'll go over this a little bit more in a few slides when we talk about our TBI patients, but it's an area that only seems to really exist in primates. So you would be able to study it in macaques. It's not possible to study chimpanzees or great apes, but they would have a similar frontal polar cortex. If you look at their tissue post-mortem, it's a similar size relative to the overall brain volume as the human, but does not exist in rodents and does not exist in honestly most primates. So things like marmosets don't really have much of a frontal polar cortex. And so these animals that have really more complex behavioral repertoires seem to have evolved more cortex in this lateral frontal polar region, part of Brodmann area 8. And this selectively encodes the value of exploring something novel. And as you'll see in a minute, when it's lesioned, we see a selective increase in apathy in our TBI patients, suggesting to me that this is a really important part of human behavior. The ability to see into the future, predict what might be valuable down the road, seems to be a really uniquely human or at least higher order primate ability. But that's sort of just a hint to what I'll get to in a second. What we went to after we did those explore-exploit studies in healthy controls is the desire to use this framework for understanding the neurocomputational basis of apathy in TBI. So you'll remember we did a little bit of lesion mapping work to understand what brain regions were damaged in folks that had apathy. Now we know in healthy volunteers the neurocomputational bases of these motivated decisions in the lab. Now we want to use that computational framework to understand what's driving apathy, specifically to resolve, is it a motivation to exploit something valuable? So do they just not care about getting points in the first place? Or a motivation to explore novel and potentially uncertain options? One other thing under the hood that I'm going to mention quickly here, because we have some data on this from this trial as well, is effort. So with our three-armed bandit task, it's always just a button press. There's no real physical or cognitive effort involved. But obviously, when it comes to understanding the brain basis of apathy or amotivation, you have to also potentially dive into the influence of whether or not you're willing to exert physical or mental effort to get an outcome, which we look at as well in this trial. So I'll mention both things. So this is all part of a Cobre Junior PI project that I worked on up at UNM HSC campus. And it's started in January 2020. So a couple months into the project, we had an interesting hiccup with the global pandemic shutting down all human subjects data collection. So the fact that there's data here is just thanks to lots and lots of people being dedicated and hardworking and continuing the work despite those challenges. But here it is. So we have, first off, our apathy plot just to show you that the clinical presentation is indeed there. So we had about just under 40 TBI subjects in the sample and just over 20 control participants in the sample. And we see that those TBI participants had significantly increased apathy across all dimensions of apathy, but primarily driven by increased behavioral apathy. So this would be reduced motivation to initiate behaviors like going back to work, getting out of bed in the morning, just can't be bothered to sort of initiate those things like they used to be pre-injury. Those scores are higher in our apathy group compared to our controls. So clinical presentation is there. We had them do our bandit task and they performed similarly to healthy controls. So this is just the fit between our novelty bandit task performance and the computational model for healthy controls and TBI patients. There obviously are some folks who were a little bit far away from the model fit, but they were still all sort of in the positive range. And I tend to, in these cases, try to include all my participants and just use robust statistical models. So these are all still thrown in the data. The main thing to point out is that these two effect sizes are not different. So the correlation between the model and behavior was the same in control and TBI. The main reason to point that out, it's not as if brain injury full stop disrupted behavioral performance on the task. It's specifically, as you'll see in a bit, scaled with apathy and TBI, but brain injured participants could perform this task totally fine and right in line with healthy controls. Now, just a quick tangent into effort-based decision-making. So like I said, we didn't want to just do our explore, exploit decision-making task because it misses this whole dimension of motivation and decision-making related to physical or mental effort. So we collaborated with Masood Hussain on a task that he's developed called the Apples task. It's a relatively straightforward approach. Basically, you have a grip dynamometer that you have to squeeze as hard as you possibly can for 10 seconds, which is it's a long time to do maximum voluntary contraction, actually. Your hand gets pretty tired. And we train people to squeeze that grip dynamometer as hard as they can. And then we give them a series of value effort trade-offs. And those value effort trade-offs will be things like, would you be willing to squeeze at 60% of your maximum contraction for 1 point? Or 20% for 6 points, or 100% for 10 points, et cetera, et cetera. And what we look at with this paradigm is how much people devalue a reward based on physical effort. So we see how likely people are to start rejecting rewards as the physical effort goes up on the task. And nicely, the plot of the behavioral data really accords well with what we would expect to see if somebody's being quote unquote rational on the task. So effort level is color-coded here. And on the y-axis, we see the probability of accepting our offers. And on the x-axis, the value of those offers. So when effort is really high, you need lots of points to be available to say yes. When effort is really low, in blue, you're willing to say yes regardless of how many points are on offer. And we can actually get the random intercept between these value and effort, this value and effort level interaction for each of our subjects to determine how much they devalue those rewards based on the amount of physical effort involved on the task. So we get both our explore exploit parameters from the model that I mentioned previously, as well as an effort-based discounting measure from this task. And I'll put them both on this slide relative to apathy to see, does immediate expected value predict apathy? Does future expected value predict apathy? Or does effort-based decision-making predict apathy, or some combination thereof? So first up, the effort one does predict apathy. So on the y-axis here, we have how much people devalued a reward based on physical effort. And on the x-axis, we have the amount of apathy associated with the individual. And we see this negative correlation such that they devalue rewards more based on physical effort, the higher their apathy scores are. But interestingly, I present all the model coefficients here to show you it's not an interaction by group. So it's just apathy scaling negatively with effort discounting, whether you've had a brain injury or not. So this is kind of independent of the brain injury. Individuals who have higher apathy scores are going to be less likely to be willing to exert effort for rewards. So that one's not all that surprising. It fits nicely with a broad spectrum of amotivation and effort-based decision-making stuff from the Parkinson's literature and other patient groups. The cool thing here that I think might be unique to the TBI patients we sampled was that there is a significant interaction in our explore-exploit contrast and apathy. So negative values on the y-axis here from our explore-exploit task indicate less willingness to explore novel, potentially uncertain options, and higher values indicate greater likelihood of exploring novel options. And what we see is that those individuals who did not want to explore new things and kind of wanted to exploit what they already knew had higher apathy scores. And individuals who had an increased tendency to explore novel things did not have as high of an apathy score. Suggesting, and there's a clear interaction by group here where there's no relationship whatsoever in the non-brain injured control participants. Suggestive of apathy in TBI specifically being related to a reduced motivation to explore potential uncertain options that might be rewarding in the future. Now let's look at the brains of these individuals, and you can see why I'm interested in the frontal polar cortex a little bit more here. So we are looking here at a brain-wide map of a signal called reward prediction error, which is just the difference between what you expect to get at feedback and what you actually do receive. And we see just proof of concept here. So areas that are involved in motivation across decades and decades of neuroimaging work, like the nucleus accumbens and the amygdala, anterior insula and the rest of the salience network, ventromedial prefrontal cortex. All of these regions seem to encode reward prediction error across the whole sample, but we have a unique negative scaling of reward prediction error in the frontal pole as a function of apathy in TBI, suggestive that damage to neural circuitry that is anchored in the frontal polar cortex, that is important for determining the future value of exploring something new, seems to be driving some of the amotivation or apathy that we see in TBI patients. So that seems to be what's driving those TBI-specific amotivation changes. We also looked at the effort discounting parameter from our apples task and scaled it with apathy. And we find that dorsal striatum, specifically the putamen or the dorsolateral striatum, seems to negatively scale with apathy and TBI as well, suggesting that this area, which is important for maintaining effort on effortful tasks, is blunted in its recruitment in folks who have higher apathy with TBI. So we have frontal polar cortex seems to be scaling with this explore-exploit deficit, dorsal striatum, and specifically dorsolateral striatum seems to be scaling with this effort-based decision-making deficit and apathy, or sorry, in TBI. Interestingly enough, these two are not related to one another. So when I look behaviorally or at neural recruitment of those regions, there is no correlation between them, but they both independently scale with apathy, which potentially suggests independent mechanisms of amotivation after a brain injury, which could be a potential path towards more precision medicine for amotivation in these patients. If you have something you might want to target to ramp up effort or willingness to exert effort, or something else you'd want to try, like dopaminergic meds to increase the motivation to try something novel, because dopamine increases novelty seeking, they would potentially be titrated based on how they perform on these two different tasks. So something that we definitely want to follow up with there. Linking it back to our models of the healthy brain making explore-exploit decisions and what we understand now with the brain injury study, the general pattern of results, which accords with models put forth by other folks in the neuroscience community, is that there is this ventral circuit anchored in ventral striatum and amygdala, as well as ventromedial PFC, that seems to compute the value of a stimulus or a goal, whether it's a goal to explore or exploit. This seems to positively encode that value signal. And then there's a dorsal circuit anchored in dorsolateral PFC, dorsomedial PFC, as well as dorsal striatum, that seems to encode the value of the goal-directed action itself. That is also accounted for effort level, task structure, etc. Then there's this last really interesting piece that the frontopolar cortex seems to be uniquely involved in encoding the value of exploring something novel, which is really just interesting to fit into the larger literature on this region, which seems to be involved in encoding counterfactual choices. So if you make a decision, it seems to encode the value of alternative courses of action in some laboratory paradigms. And in our hands, it seems to encode the relative future value of exploring something novel. And it's a primate-specific region, suggesting it's important for complex, sort of sequential or hierarchically organized cognitive tasks. The main thing I want to point out here is that we explore for a reason. I think lots of folks view novelty seeking and sort of hyper exploration as potentially problematic, but the fact that this really higher order frontopolar region encodes the signals necessary for it suggests to me that it's got a really evolutionarily significant purpose for humans and other organisms, and also developmentally significant. So we are now looking at explore-exploit decision-making in the context of adolescent and emerging adulthood development. My sister will kill me because I use this as an example now in my talks. This is her wall from our house when we grew up. And as you can see, her interests varied. So I have pictures of Method Man alongside Mark Wahlberg for some reason. I'm assuming if that was still up, she'd want to take that one down now. Lauryn Hill, Radiohead, Bill Clinton's on there, just Toronto Maple Leafs hockey team. A really varied assembly of interests that she explored over the course of her adolescence and emerging adulthood. And the reason I'm showing this is because I happen to know that after this odd obsessive period that she went through during her adolescence, she became an extraordinarily well-adjusted adult with very clear preferences for some of these things, but not all of these things. And that sort of demonstrates the general cognitive profile that we expect during adolescence and emerging adulthood. You bounce around and try lots of things, you learn who you are, and you develop preferences that end up shaping your personality as you become an adult. And what we're doing now with the learning and exploration in adolescence study, or the LEAD study, is we're studying how the human brain starts to acquire this ability. How we go from what we refer to in the field as random exploration, where you're just kind of bouncing all over the place, whether or not there's sort of a formal value of information associated with a novel cue, versus directed exploration, where you're exploring new information, and as the value of that new information decreases, so does your tendency to choose it. How do humans transition from doing more random exploration to directed exploration in that adolescent to young adulthood period? And what we expect to see from a developmental profile is essentially random exploration will be kind of constant. So during early adolescence and middle adolescence, random exploration will probably remain high. And then even in adulthood, it would kind of still remain high through like inattention or other reasons driving it. Whereas directed exploration, we expect to see a maturational shift. So basically at some point during adolescence and the transition to young adulthood, we expect people to become more strategic or goal-directed in exploring novel options. And the main hypothesis of this work, it is funded by the NIAAA, is to see whether or not lagged or absent maturation of this process relates to increased substance use challenges early on in life or during that adolescent to young adulthood transition. Now, obviously, this is a big time period to study. It's not a 20-year R01, it's a 5-year R01, so it's actually a cross-sequential cohort design. So we're going to have 135 folks come in, starting off at 13 to 21 years old, and each of them will come back for 3 time points with the goal of sort of filling in this entire longitudinal trajectory in a 5-year period. I don't have, so this just started this year, I don't have the longitudinal data for you yet, but now you're going to get a sort of first peek at some of the data that we're collecting cross-sectionally. One other note before I show data is that we're doing both the bandit task in a win context, where you're trying to maximize wins, as well as a loss context. One of the other things I was noticing about our prior work here is that we had kind of just disregarded the fact that people are often exploring in order to get away from bad decisions. So we have both of these contexts tested in our study, both exploration to gain and exploration to avoid loss. And interestingly enough, when we look at those two situations, so maximizing reward or avoiding loss, and we look at explore-exploit decision-making across time, we see a really clear difference in exploration upfront. Already, we're at n equals 43 right now in this trial. And the loss context is clearly boosting the motivation to explore something new. So essentially, when you're in this context where you're trying to avoid losing, you're way more motivated to try out the novel options than you were in the win context, which is already an interesting finding that we kind of hypothesized. So hopefully it holds once we get all of our data in. We're doing the same computational modeling work in this study. So we have our immediate expected value, or IEV parameter, and our bonus parameter, plotted here by loss and win context. And we see that basically the bonus value significantly increases in the loss condition relative to the win condition. These are individual data points plotted with the ball and stick lines as well. Basically, an increased valuation of uncertainty seems to exist when you're in a loss context relative to a gain context. Last thing, as far as new data that I'll show, is the association between these parameters and age. So these folks, like I mentioned earlier, are from 13 to 21 years old. We only have 43 folks cross-sectionally. We're going to have way more to look at this later. We're also going to have longitudinal data to look at individual change across time. But what we're finding so far is that the association with age is specific to the win context and the motivation to explore uncertainty. So when it comes to that strategic directed exploration increase during this developmental period, we seem to specifically see it in the context of wins and with respect to that bonus parameter, not the tendency to exploit something familiar. So the hope now is that this will predict some specific clinical outcomes in this trial, the main one being early adolescence and mid-adolescent alcohol use. And that was predicated on a recent finding where we used our same bandit paradigm, but with alcohol or non-alcohol cues, and had people doing the same explore-exploit trade-offs with alcohol or non-alcohol stimuli. In this trial, we had hazardous drinkers versus controls. And we basically found that the hazardous drinkers were way more motivated to explore novel alcohol cues relative to novel non-alcohol cues. And our basic working framework for this is that at this stage, the preferences have been developed. They prefer the alcohol stimuli, so they're more motivated to explore it. In our trial looking at adolescence and emerging adulthood, we're hoping to understand whether noisy or random exploration patterns that persist into sort of mid and late adolescence are predictive of the early tendency to develop these preferences. And so we're going to look at alcohol use outcomes in that trial as well. We have a lot of them measured, and we have folks who are like Katie Wickwitz is working with us on this to figure out how to analyze that data. So hopefully we'll have more to say about that soon. At this stage, we've just done a lot of the analysis of the choice data and are moving on to the clinical stuff next. Last thing to mention, we are interested in using this to modulate brain circuits involved. So all of the work that I've mentioned so far is all well and great from a neuroscientific perspective, but might not be super helpful from a clinical perspective if we can't use it to help people. So what we're doing in a recent trial that we collected some pilot data on is using our analysis of these motivational neural circuits to guide individualized neurostimulation with transcranial magnetic stimulation, where we pick individualized targets for each person. These data are at the top of my list to analyze right now. I will hopefully have more data on this to show you soon as well. But just to give you an idea for where this is all going, we want to do this computational neuroscience work to understand mechanisms, development, and pathology, all in order to potentially guide precision modulation in the future to maybe develop treatments that help people with mental health challenges. So it's not all just to understand mechanisms. I'm really interested in helping clinically at some point in the future as well. That is it. I think I ran like a minute long. Sorry for rushing down at the end. I wanted to make sure I got done with some time for questions. Thank you all for your attention. Any questions?
Hi, yeah, thank you. My name's Harrison Grace, one of the child adolescent psychiatry fellows. Can you hear me okay?
Yes, I can. Hi, Harrison.
Thank you. Yeah, fascinating talk. And as a brief disclaimer, I have a PhD did an MD PhD in neuroscience too. So this
Oh,
tingles a
cool.
lot of long dormant research interests. But that aside, one thing that kept popping out to me hearing about your studies, your results, your plans here is very relevant to what I see a lot in adolescent psychiatry.
Hmm.
As you mentioned, these decision-making processes important in normal adolescent development, and part of that whole stereotypical adolescent phenotype, but also the effortful choice and motivation of novelty versus known reward strike as very kind of topical things with ADHD, attention deficit hyperactivity disorder, and
Right.
curious if there's been applications of these methods into studying that.
Yeah, no, so there are the general like probabilistic reinforcement learning framework that our task kind of is based on. There's definitely been studies of that in ADHD, and they typically find sort of things like a slower learning rate, a probabilistic learning rate on an RL task, and that if you give folks with ADHD stimulant medications to treat the ADHD, you tend to see like a sort of recovery or return to a normal range learning rate. I don't know if anybody's done the explore exploit work specifically in ADHD though. So that's one that we are going to get. So there's some folks already that we've recruited in our task who we're not necessarily excluding for stimulant medication use. We kind of want to see like what we kind of want to include everybody in that adolescent age window to get like a general idea of what might be moving the needle on some of this stuff. And so we've definitely had some people come in who have diagnosed ADHD or at least are being seen for ADHD. So I will hopefully have more to say on it soon.
Oh, really cool.
Yeah, but no, I think it's certainly in my mind when it comes to like random exploration and sort of not learning optimally to exploit, I think that would be the end of the continuum that I described that I would expect to see related to ADHD phenotype. But yeah, TBD.
Yeah, nice. And for whatever worth my 2 cents here, I think that your approach you just mentioned sounds like a good idea of not being too strict, excluding or including, because it's at times influx diagnosis in the process of
Yes.
normal adolescent development. So cool.
Yeah, for sure. And I think we just try to get as much data as possible from everybody that comes in. So we'll know everything about whether they've had a diagnosis, and we'll have lots of neuropsych to evaluate their attention processes and stuff. And hopefully once we consider all that, we'll be able to really sort out some signal from noise there. But yeah.
Cool. Well, thank you again. Very fascinating stuff.
Oh, thank you. Davin, you got a hand up?
Yeah, there's a question in the chat that I think popped up
Oh,
before me, but I'd love to ask the question after that.
sure.
I'll read the question in the chat for you. It's
Okay.
a common clinical observation that TBI results in impulsivity. Your research shows increase in apathy by different mechanisms. How do we reconcile those?
Yeah.
I'll stop there and then read the second question.
Yeah, no, that's a really good question. I think similar. I remember when I worked in an autism center as a postdoc, and one of the most insightful things somebody told me was that if you've met one kid with autism, you've met one kid with autism. I think TBI is a little bit of that type of diagnosis as well, where depending on lesion location and a lot of the other variables that were existing comorbid or before the lesion, there's a lot of tendency for really heterogeneous outcomes with TBI. And that is very true with respect to impulsivity versus apathy. So there will be varying levels of impulsivity and apathy within an individual that has had a TBI. So there can be context dependent. They might be apathetic about going to work or getting back to their old friend group, but really impulsive when it comes to new behaviors that they've developed post-injury. So sometimes gambling disorder or substance use can be linked to TBI. So I think you can have sort of context-dependent shifts of that continuum between apathy and impulsivity. But what I've seen as far as like interacting with patients that come in for studies, and particularly I saw this a good amount at the Rehab Institute of Chicago when I was postdoc there. There is definitely some people that are more on one end or the other as like at like a trait level after an injury. So more impulsive or more apathetic after the injury, just globally. And I think I don't have the statistical power yet to resolve what would drive somebody to have one versus the other phenotype. I just definitely can agree with you that both exist and are probably some combination of related to different different neural circuit damage, as well as different premorbid function that causes different paths after the injury, as well as the environment and the social environment post-injury. So I think the answer to that one's probably going to be complex, but definitely can have both phenotypes. I agree with that.
Maybe real quickly, I'll read the second question, and then Dr. Quinn has a question. Is your research more focused on targeted or goal-oriented decisions like burger versus sushi?
So, I mean, targeted or goal-directed, I would view as kind of the same thing. I'm not totally sure I'm understanding the question there.
Yeah, that was a continuation to the same question, because if it is more targeted behavior, then it would make more sense to see more empathy. But if you're talking about decision making in general, it would make more sense, at least to me, that TBI patients would be more impulsive. But
Right.
you already answered that, so thank you.
Yeah, no, I think it's totally possible to have like state-based shifts or context-based shifts between those two. But then there are definitely some patients that you see who it's like this person is just going to try everything and is not necessarily controlling their behavior like they might want to. And then others who have sort of a global loss of motivation after the injury. And that both of those are kind of the type of motivational deficit I'm talking about here. But yeah, we need more data to really solve that continuum, I think. Davin?
Thanks. Well, I do have to say, hearing you talk is always a good antidote for my own apathy. It just gets me so about where your stuff is going and what we all can do together as a neuroscience community. So thank you for presenting this. It's awesome to see.
Thanks,
and my question is about do you find like any laterality sort of in your stuff? Is left side versus right side implicated more on these in these circuits? I might have like a few hypotheses about if one were the case, or is it bilateral symmetric? I'm just curious if you saw any sort of lateralization of these phenomena on your image.
All I could say is in the healthy brain, I always see things bilaterally on these tasks. So that's actually like one of my typical, we call them positive controls in the lab. Like you look for the thing that you should see. Like if you're responding with a right hand, then we should see left motor cortex. And when it comes to our motivational signals, it should be bilateral for most of them in the healthy brain. That being said, I think I have to look at the map to remind myself, but I think for the thing that's scaling with apathy and TBI, it's very left lateralized. And I think I need to do some more. So we have like perfusion data from these folks. We have DTI data from these folks. We have lots of different potential signals to look at the mechanisms, whether it's perfusion related or structural, that could be driving these deficits, to see whether or not maybe more of them had left prefrontal cortex damage, and those are the ones that are really driving this, or maybe it's not related to the local damage and it's a network related change that everybody shows. Those types of things I still need to sort out. But yeah, the frontopolar cluster that scales with the reward prediction error response in our task is left lateralized. I'm almost certain. I would need to go back and look at the plot again. Yeah, but bilateral in the healthy brain, I will say.
Thanks so much.
Yeah, no worries. Any other questions? Okay.
All right. Thank you so much. Look forward to seeing the next group of data as well. There's a lot of directions you are going in. So this is going to be great to see how this evolves.
Thanks so much. Thanks everybody for attending. A pleasure.
Bye-bye.
Have a good Friday.